So to date, new technology to detect antimicrobial susceptibility has really taken one of two approaches. So we can look for a gene in an organism that's thought to predict its resistance to a given antimicrobial, so that's a genotypic approach, or we can do a phenotypic method, which answers the question, if I incubate this bacteria with this drug, does it prevent it from growing, or is the organism resistant and can grow even in the presence of the drug? And so both have advantages and disadvantages. To date, most technologies have really focused on genotypic approaches, and so there are some challenges with that, in particular for some organisms that we have real problems with today, like the gram-negative bacteria. And so by looking for just a single gene, you don't get the entire story of what's happening in that bacteria. And we know in gram-negative infections, resistance is usually multifactorial, and a combination of the presence of a gene, its expression, as well as other factors like porins or efflux. And so by asking, is that gene there, you don't really know what else is going on. And so when you detect a gene, you must assume resistance. But we know that often the gene will be present and there will be no resistance. And so this is one big challenge. You're not really able to detect susceptibility using a genetic approach. You're only able to assume resistance. The other challenge with it is there's a disconnect between what we use called the clinical breakpoint versus a genotypic method. So when you're doing a genotypic method, you're looking to see if the organism is wild type, natural state, or if it's acquired a resistance mechanism. But that doesn't necessarily mean that it will have an MIC that's above the clinical breakpoint. And so you still may be able to treat some of these infections even though they have a resistance gene. And we've seen that actually numerous times in our lab. When we've started to do things like whole genome sequencing of bacteria, you find a lot of resistance genes, but either they're not being expressed or they on their own don't contribute to a resistance phenotype. And so for these reasons, I really think that having a phenotypic approach is a lot more desirable. However, you need an awful lot of bacteria to do a phenotypic approach, and it also needs more time than we really want to spend waiting for a result, because you need to give the organisms time to respond to the antimicrobial. And so, those two things combined make it very difficult to make a very rapid, direct from patient specimen phenotypic test. Nonetheless, a phenotypic test does not matter which mechanism is causing the resistance phenotype. And also, as we have new antimicrobials come to market, and we don't know what those resistance mechanisms will be, a phenotypic approach should be able to detect resistance or susceptibility in those cases.